In 1957, in a London laboratory, two scientists were working with pieces of chicken egg membrane. They exposed these cells to a heat-killed influenza virus, a virus that was completely inert and incapable of infection. Then, they tried to infect those same cells with a live, active virus. But something strange happened. The second virus couldn't take hold. It was as if the dead virus had left behind a ghost, a protective presence that warned the cells of the coming danger. They didn't know what it was, only that it was some kind of protein. They called this mysterious substance interferon for its ability to interfere with a viral attack.
This interfering factor wasn't a weapon itself, it was an alarm bell. When a single cell in your body is breached by a virus, it begins to scream for help by producing and releasing these interferon proteins. These proteins pour out into the space between cells and travel to their neighbors, acting like messengers carrying a vital warning. They dock onto specific receptors on the surface of nearby healthy cells, essentially knocking on the door to deliver the bad news. This single event triggers a cascade of thousands of signals inside the recipient cell, commanding it to enter a defensive antiviral state.
What does it mean to be in an antiviral state? It means activating hundreds of otherwise dormant genes. One of the most important is a protein called protein kinase R, or PKR. PKR is a molecular scout, constantly scanning the inside of the cell for signs of an intruder. Its target is double-stranded RNA, a structure rarely found in our own cells, but is a dead giveaway for many replicating viruses. Upon finding it, PKR activates, and its sole purpose is to find and shut down the cell's ribosomes, the microscopic factories that build all proteins. By shutting down protein production, PKR not only stops the virus from making copies of itself, it puts the entire cell into a state of suspended animation. But freezing the cell isn't the only option. The interferon signal also wakes up another sentinel, a protein system called OAS. Like PKR, OAS is triggered by the presence of viral double-stranded RNA. But instead of pausing the cell, it initiates a self-destruct sequence. Activated OAS triggers a second protein, RNAL, which is best described as a molecular wood chipper for genetic material. RNA begins to indiscriminately destroy all RNA it can find within the cell, both viral and cellular. It's a scorched earth tactic. The cell commits suicide to ensure the virus cannot spread. The defense isn't just internal. Some antiviral proteins work on the cell's outer border. One of these is called tetherin. Imagine an infected cell has become a virus factory, and newly assembled virus particles are trying to bud off from the cell's surface to go infect others. Tetherin acts like a microscopic leash. As the new virus pushes its way out, one end of the tetherin protein stays anchored to the cell membrane, while the other end latches onto the escaping virus membrane. The virus is trapped, physically tethered to the surface of the cell it just came from, unable to float away and continue its spread. Of course, for every defense, there is an offense. Viruses have been co-evolving with us for millions of years and have developed their own sophisticated countermeasures. The influenza virus, for instance, produces a protein called NS1 that directly binds to and neutralizes PKR, keeping the cell's protein factories running. Some herpes viruses create decoys to distract RNase L, while HIV produces a protein called VPU, whose specific job is to find tetherin and destroy it, clearing a path for the virus to escape. This arms race has led to even more elaborate defenses. Your cells contain a family of proteins called TRIM. One of these, TRIM5, doesn't wait for replication to start. It intercepts certain viruses, like retroviruses, the moment they enter the cell. As the virus sheds its outer layer, it exposes its inner core, called the capsid. Multiple TRIM5 proteins immediately recognize this foreign structure and begin to spontaneously assemble around it, forming a hexagonal, cage-like lattice. This cage effectively quarantines the virus's genetic material, preventing it from ever reaching the cell's nucleus. The invasion is stopped before it can even begin. This raises a fundamental question. If your cells are armed with these sophisticated alarms, brakes, shredders, and cages, why do you ever get sick? Part of the answer is a simple matter of numbers. A sufficiently high viral load can overwhelm these defenses before they have time to fully activate. The speed of viral replication versus the speed of the interferon response is a critical race. But it also suggests that the effectiveness of these systems might not be the same in every person. Your internal cellular battle against a virus might be very different from the person's next to you. We know that some people are naturally resistant to certain viruses. The classic example is the CCR5 delta 32 mutation, a genetic quirk that removes the docking port HIV uses to enter cells, granting a high degree of immunity. But what if there are other, more subtle variations? What if someone's DNA codes for a version of PKR that's slightly faster, or a TRIM5 protein that's better at building cages? Scientists are now hunting through the vastness of the human genome for these exact variations, these gain-of-function mutations in our innate antiviral arsenal. The idea that natural immunity isn't just something your immune system learns, but something your cells are fundamentally born with, changes everything. It means the blueprint for surviving the next pandemic might already be written inside someone's DNA.
Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.